My name is Kim. I'm a biologist at the BioInnovation Institute, working for Neurophase Therapeutics. Why is there this weird buzz? Do I have to do something else?
I'll try to touch. Go ahead.
Okay. First of all, I would like to thank the Shine Foundation and everyone who's here for inviting us, for giving us the opportunity to talk here. We are a very young startup, and it adds great value to our work to be so connected to the patient community. And I think as scientists, we sometimes lose a little bit touch of the real-world problems. We go into the labs and we're really sciencey and nerdy and techy. But this experience here is really grounding and humbling, and I really appreciate that. So thank you very much for that. During my talk, I will introduce biomolecular condensates as a novel drug target for treating neurodevelopmental disorders. Before I dive into the research, I just want to explain a little bit the framework of Neurophase Therapeutics. Who are we? What is bioinnovation actually? And I think for you as a patient community, that's important to understand what drives our work, what's our catalyzer. And for you as scientists, it's also when we potentially collaborate together to understand what are we actually. I will start with the BioInnovation Institute, short BII. It was established in 2018. It's founded by the Novo Nordisk Foundation, but we are really independent from Ozempic and all that research. We are just founded by them. We are located in Copenhagen, Denmark. And you can imagine it that BII is like a startup incubator. So in the house, we have around like 40 startups currently. And we have, like you can imagine it as a one big community. We are sharing facilities like the office and lab spaces, resources, knowledge. It's very interactive. All the startups, they're science-based, and we have 3 focus areas, like human health, planetary health, and societal resilience. And at BII, our mission is to bring science to life by identifying unmet needs and supporting early-stage startups that will create long-lasting impact. And for us as Neurophase Therapeutics, obviously we are situated in the human health, and we're also pursuing the high unmet needs, which you obviously would know. And that as a startup, as a young startup, that's also a risk-taking. When we started like 1.5 years ago, we had like a patient community of around about 100 patients. Now it's double, more than double. But as a startup, that's kind of a risk that we're also taking. But this is what BII is doing. BII is at the heart of all the startups in the house, and they're supporting us with the infrastructure, the network, the knowledge, the financing, and the overall business development. So many people in BII have an entrepreneurial background. We are collaborating with universities, hospitals, and people from industry, and bringing them to 3 different programs. And this is BioStudio, Venture Lab, and Venture House. And these different programs, they differ in their maturation level. And our Neurophase project is situated in the BioStudio, and that is where all the early stage startups are starting. And to understand, we are not yet a company, but we have 3 years of time to create a company and spin out a startup at the end. So basically, we're a startup project, so to say. Just for BioStudio, the model, because that's quite unique. In the BioStudio model, there's 2 research institutions basically involved. It's an academic institution, and then there's also BII. And we have an official, or BII has an official collaboration agreement with the University of Copenhagen in our Neurophase case. And we are particularly having a collaboration with Professor Christian Strømgaard. And some people might know him from Avilex already. So to understand, our science that is underlying Neurophase Therapeutics is originated in the research group of Professor Strømgaard, Christian. And BII has given our project a soft funding for 3 years. And we have a team at the BioInnovation Institute, and that is led by an entrepreneur in residence, a manager, so to say. And we are conducting the research and maturing it at BII, and then hopefully at the end of the 3 years, we are spinning out a startup successfully. This is the team and the people all involved in Neurophase Therapeutics. It's quite a lot. So first of all, we have the research group from Christian, of course, and also his PhD students, Elena and Tobias, and then also master students working on this project. In Neurophase Therapeutics, we have 2 postdocs from Christian's lab, and this is Christian Kolby, and then also Vita. And Christian Kolby right now is the interim EIR, the interim manager, but we're also hiring a new manager right now. And then for the drug development, there's also Jaroslava together with Vita. And then there's me as a biologist and also Alberta as a biochemist. And then we're also collaborating with other advisors and academic collaborators of scientific and business nature. And one person to mention here especially is Mingji Zhang, Professor Mingji Zhang. He's basically the person or the scientist who has identified the phase separation of our protein complex in neurons. And Oren Davinsky is also, many people might know him from his epilepsy research. And then, of course, Zainab, I don't need to introduce her, and Jonathan, and then also business advisors. Just to give you an overview where we are, basically. So we are right now half year through, so in 1.5 years through. And right now we are basically at the proof of concept stage. And there's many things that have to be done in a startup journey. It's not only the science and the data, but it starts with the IP definition, the fundability, the financing later with the VCs, and also the whole business development. For you, of course, it's always interesting to see where are we in the clinical stage or are we preclinical. I will tell you we're definitely, of course, early preclinical stage, but I will guide you through what we have so far. Just as a quick wrap up. So Neurophase, we are an early stage startup. We are having a novel drug target for neurodevelopmental disorders, biomolecular condensates, and we are following up on a peptide-based drug discovery approach. And that is something that is maybe a little bit new. Many people know gene therapies and also small molecule approaches, but this is why I just want to highlight some benefits of this. So peptides can mimic natural ligands, and they're less immunogenic, so they might not cause such an immunosuppressive response. And they have a high selectivity and affinity and less off-target effects. And so overall, there might be like an improved safety, because once the peptides are degraded, they degrade into amino acids, and these are naturally occurring and less toxic. Just to highlight that. But let's talk about the biomolecular condensates. I will take you out of the laboratory and take you into the kitchen for one slide. And I think everyone has done the observation. If you pour oil into water, the oil will form oil droplets and separate from the water. And that is something very simple observation. And you just can take that and transfer that to proteins as well. So in the cells, you have proteins floating around. And these proteins, they can form protein-protein interactions, and via a process that we call liquid-liquid phase separation, or short LLPS, they can form these condensates, basically. It's a little bit like protein aggregates, but what is the difference is that it's a highly dynamic process. So they can go from the condensate state back into the dissolved state. And that can be a very highly flexible process. And there are multiple examples for condensates, like the nucleoli or the germ granules, for example, stress granules as well, and also the synaptic densities, like the postsynaptic densities that we have heard a lot about today. And actually, that's a novel way of looking at the cell, because how we also call these biomolecular condensates is membraneless organelles. So basically, when we're coming from a classical cell organization, we think as scientists of membrane-bound organelles, like we have the nucleus, the mitochondria, some know them as the powerhouse of the cell, and they're all surrounded by membranes. But when we talk about the membraneless organelles, this is something like a novel way of looking at cellular organization. And one very important study for our work was published by our collaborator, Professor Zhang. And he, for the first time in 2016, showed that proteins, especially the PC95 and the SYNGAP complex, they undergo this liquid-liquid phase separation. And that was quite a milestone for our work, basically. To understand. So these condensate dynamics, our hypothesis is, and it's a very strong hypothesis, is that brain disorders or mutations from patients, they could interrupt these dynamics from the condensates, and it can be shifted in any way. So maybe it can be that the protein interactions, they harden the condensates, or it can also shift into the other direction, that these condensates are more dissolved. And we believe that mutations from patients can interrupt these dynamics, and this is exactly where we want to target. Basically target the condensates with our peptides and see that we can modulate it into the right way again. And we're looking at the most abundant and most important proteins at the postsynaptic density. That's PC95, SYNGAP, GCAP as well, on Schenker and HOMA, and they're forming this scaffold that we have heard a lot about today. So to understand how we're approaching that, so for this we're using the most simple approach, an in vitro approach. And it's basically very, very simple. We're just purifying and synthesizing the proteins like PC95 and, for example, also SYNGAP. And we're pipetting them together, mixing them up in an Eppendorf, and then put them under the microscope and see that we can, and we also tag them with the fluorophore, and then we can see these droplets. So this is something where we change the different concentrations. It's a little bit like a phase regime, but you can see that different concentrations also lead to less or more phase separation of these proteins. And this is also where you can see it, like basically when we do it in the microscope, we mix them together, we put them under the microscope, and they form these condensates, and then they fall down because they're a little bit more heavy and like sedimentation. And this is basically what you can see here. And you can do this with all the PSD components. So this is one of the first experiments to show. And here, because many patients suffer from haploinsufficiency, so they have half the amount of protein, basically. And this is what we wanted to test it in the in vitro system. So we tested like an in vitro haploinsufficiency model. So what we did is that we mixed the different components of the PSD, like the PC95 protein, the SYNGAP, the GCAP, and also parts of the NMDA receptor, mixed them together at same concentrations, like 5, 10, or 20 micromolar, and the only concentration that is changing then when we're adding the PC95. So that's an increasing amount of PC95 what we're adding. And what we can see is that PSD95 is the limiting factor in the PSD phase separation of the core PSD95. So if you can see that here, it forms, when you add PSD95, there's more condensates that are formed. And if you would add something else than PSD95, that is not what you would see, basically. So basically, this shows that mimicking PSD95 haploinsufficiency in the in vitro reduces the phase separation. And then there's another important factor, and these are the missense variants, and I think most of the people are also interested in those. And Dr. Thümer has already shown a little bit of this. And here we tested the phase separation properties of missense variants. So we have the PC95, the PC95 wild-type protein, and then we have two different missense mutations that we have introduced. And then there's also another one where we have deleted the PDZ1 and PDZ2 domain. And you can see the mutations here. So basically, what you can see is that on the axis, there's the phase separation in percentage. And when you when you look at the mutant B, you can see that there's a decrease in phase separation compared to the wild type. And also the mutant A. And we would say that a mutant A, there's no effect in phase separation, but in mutant B, it looks like there's a decrease in phase separation. And also this one shows where we have deleted the PDZ domains 1 and 2, that these domains are important for the phase separation. So both of these results are quite important for us, from the haploinsufficiency system, but also from the missense variance, because it can show that there might be a juiced condensate phase transition, basically, or formation. And that for us is important to know, because then we know we need to drive the condensates formation, basically, with our peptide approach. Yeah, then also to I think this is also what Dr. Tuma also already presented a little bit, like as a short overview, but we also tested the effect on protein binding and stability from the missense mutations in PSD95, the same that I showed before. So this is, the first one is a protein stability assay. And you can see that the lower it is, that just means the protein is unfolded. And for the mutant B, you can see that this is very unstable. And the mutant A as well is also a little bit unstable. And then, due to this, that just means the protein is not folding correctly anymore in that specific region. And then we did a binding assay with the primary ligands of PSD95. So we have PSD95, and we added parts of the MNDA receptor, or also SYNGAP for other domains. And what we can see is that in mutant B, there's no binding at all. And in the other one, there's in the mutant A, there's a decreased binding to the ligand, to the parts of the NMDA receptor. So to summarize this, it just shows that the mutant B of PSD95, that the protein shows a loss of its domain structure and stability, and is rendered unfolded, and therefore the protein has a complete loss of binding to its ligand, to the NMDA receptor tail. And for the PSD95 mutant A, it just shows that the protein shows a decrease in its domain structure and stability, and a decrease of its binding as well. Yeah, coming to the peptide-based drug discovery. So we are developing two different classes, because our hypothesis is that we need to drive the condensate formation so far, but we need also more data to validate that. We will also need to run more missense variants. It just takes a lot of time and work, but this is definitely on our plan to do that. But right now, our hypothesis is that we need an inducer. And we have three different classes of inducers that we have developed. And also since we still don't know if that's really what we need, we're also developing reducers of the condensates. And this is, for example, just to show you how it can look like in an experiment. This is a peptide which is inducing the condensate formation. So basically, we have some parts of the PSD components, like proteins expressed. We mix them together, and we also add the peptide and increasing concentrations. And what you can see is that these droplet formation, it increases a lot. And if we test the reducer, then it's the same basically, but we choose a concentration where we see a droplet formation and then add the peptide, and you can see that the droplet formation is diminished by that. So the current state of our startup is that we have a lead peptide so far, and it has undergone lots of optimization. And we went from a precursor peptide, basically, which did not induce so much condensate formation. You can see it at 50 micromolar. It's very weak. And went to optimized peptide. And this optimized peptide, as you can see, is a very, very potent inducer. So if we add it already at lower concentrations, you can see that it just induces phase separation. And we have done a lot of lead optimization here. These are just some examples of it. We have increased the peptide affinity, as you can see in the first one, and we also have played around with the peptide structure and tested different, different structure constructs, and also have done like the optimization, where you can see that with the optimization there's clearly a strong phase separation compared when we don't have the optimization of the peptides. Now that we have our peptide lead, it's of course also very interesting to screen it in assays. And for that, we have also established cell assays. And this is based on HeLa cells. So basically, we are just taking HeLa cells and expressing parts of the PSD, like PSD proteins in the HeLa cells. And then you can see very, very nicely that these HeLa cells, they form also these condensates within the cell. Exactly, and the idea, of course, is then to have the HeLa assays and see the condensates, and then if we add our peptides to see if they reduce or increase the condensate formation. We also already increased the complexity of the HeLa cells, so expressed more components of the PSD95, exactly where we can also then screen on future, but this is all undergoing optimization right now. Then, of course, it's also important to test the peptides in systems which are more biologically relevant, like neurons. And we started our neuronal assays. And this is work that we just have started. So this was the first shot. We are working with primary mouse neurons from the hippocampus right now. That was the first staining of it, and I think we heard already about the puncta, the PSD95 puncta. We're also using this near super resolution imaging. Maybe we're using the same microscope. Yes, exactly, yeah, I kind of thought that. But of course, we're going to dive into that and look into the structure of the neurons and also do plan for functional assays, but that will be really interesting to see. Because our lead is basically just ready, and we just want to see what does it now do in the neurons and how does it relate to the clinical indications, what could we use that for. And yeah, the last data here is just viability assays with our lead compound. So that is important, of course, to see how neurotoxic the peptide is. And we have just added our peptide to the neuron culture, basically, and then checked for the viability of how many cells neurons can survive with that. And we've done that with rat and mouse neurons as well. And you can see for the control, which is not neurotoxic at all, we know that, most of the cells just survive, but if we add the peptide, the viability decreases at higher concentrations. But this is also for us to figure out at which concentration can we work with the peptides, around like 25 micromolar, 12 micromolar, and that is in a good range for us so far to work and proceed with this lead, actually. Yeah, and also a little bit of optimization here. We've done that with the precursor peptide as well, where you can see it's a little bit more toxic, and optimize the peptide. There's not a huge difference, but you can see with small optimizations, we can just tweak it into the right direction. Yeah, the proof of concept, I mean, that is where we're at right now, and we're really ready, and the biological outlook. In future, we will extend all the neuronal assay work. We're also thinking about functional studies, also about working with EMEA, of course. I think everyone is doing it right now. So it would be super interesting, and this will come in the next weeks and months as an outlook. And we would also look to collaborate with other people. We're super interested to run that in IPCs as well. But also to run it in a mouse model. I'm looking at Sharon. Yeah, but basically our lead is more or less ready for this. We need to do the tox studies for that, but we would be really happy to collaborate on these things together. Yeah, I want to thank you for your attention and all our collaborators, and yeah, for having me here. Thank you.
Very strong finish. I have a bunch of questions. Is the peptide targeted towards PSD95? There's something on the peptide that targets it?
No, it's targeting the PSD95.
It is targeting the PSD95?
Yes.
Okay.
And it's targeting a specific domain as well.
Oh, okay.
Yeah, exactly.
What is the half-life of the peptide within a cell or within a mouse? Do you know, is it a very stable entity?
Yeah, we have tested that, and I think for the current, it was around like 6 hours or something like that.
Right. And so from a dosing perspective, as we go towards the clinic, it would be a daily dosing or something along those lines, or hard to say?
These things, the dosing, we really have to figure out down the road. But I will also say the half-life time we have tested in mouse plasma, which is known to be most aggressive.
Okay.
Yeah,
okay. And then are we talking about an LNP or some sort of vector targeting thing where you would put the peptide in order to get to the neurons, or how do you get from delivery-wise?
That will also be interesting, but this is also something we will figure out when we come there.
Okay.
But there is studies and there's ways how to do that, because one question is, of course, always how can it cross the blood-brain barrier?
Yes.
But there is ways to do that.
And just one final as I wander over, is the condensate, the oil drop, where the biological activity happens, or is it a warehouse just keeping it in supply and it's in a non-condensate form where the biology happens? Does that make sense?
We don't know.
All right.
We have to figure out.
Okay.
Yeah.
Okay. All right. Thank you. Yes.
So if I understood it correctly, and I'm speaking from, I have no idea, like you guys created a brand new molecule that you can't share just yet what it was, right? Like it from a patient that doesn't understand science perspective or a caregiver.
The peptide? Or
yes,
what do you,
the peptide.
Sorry, I'm not
the
sure.
Peptide. Like, is that the molecule? Like it's a brand new. It's not an existing drug.
Yes, exactly. Yeah.
It's a brand new molecule that you created.
This just in, it's a new drug.
Right. Okay. I want to clarify that it's a brand new, does not exist.
Yes, exactly. It's no repurposing or so.
Correct.
We started it from scratch, basically.
Correct.
And it's been optimized for some time right now. But yeah.
And then follow up to that is, will you be eventually doing clinical trials that our children would be eligible to enroll in?
Yes. I think this is something that I cannot answer because I cannot overpromise. And so far, and this is why I also showed you a little bit the funnel where we are. Basically, we are in the proof of concept. And this is certainly something that we need. And before we can file an IND and go into clinics, we need a very strong preclinical data package. And that is what we're working on right now with a proof of concept and also testing it in an in vivo model, in the mouse model, and see what it does, to really see if it can be used through all the clinical indications of the patients, because that is something that we're missing right now. But we really believe that there's potential.
Got it. And what is the timeline looking like?
Wow, that's a question. ASAP, hopefully.
ASAP. Okay.
Yeah, it's always like this. But yeah, we know how it works with the clinical trials, and to go get there, it's a lot of work and a lot of time going into that. Yeah.
In this question, I want to support Kim, because Christian Strømgaard always shows a slide for another peptide he has been created to decrease the amount of PSD95. And it is now being in clinical trial, I think, 2 or 3.
It just enters 2, I think.
2 enters 2, and he's always showing this slide because it has taken a lot of years for him to do that. And this is used in stroke, but this decreases the amount of PSD95. He has been working with PSD95 for 15, 20 years.
Yeah.
Wow.
And he always shows a slide that it can take from 10 to 15 years.
Yeah.
10 to 15 years.
It can take. But now they have the experience, and you older they have the peptide, so it kind of decreases the amount. But it may give you an idea how long it may take.
Yeah.
It's not the same platform. It's not the same.
No, it is not.
No, but the thing is that they have to use the in-cell models and mouse and everything. So that's.
You just take a wild-type mouse and check for toxicity and tolerability.
Yeah.
Do you see any? Because this seems to me the main concern now, given that you showed in vitro that there is this toxicity with increased amount. So the therapeutic window would be very, very limited, right? So do you have any insights on?
We have not done that yet, no. We still need to do the tox studies. Yeah.
Yeah. Although in my experience, the in vitro toxicity can be more severe than in vivo. We look at a drug called fluoxetine and some other drugs in neuronal cultures, and at clinical levels it kills all the neurons, but we quite clearly know it's clinically accessible, so yeah.
Yes, and actually this viability test, I also have to say, they were just released in the past weeks. Like basically I've done them in the past month. Right. I think I got my neurons 2 months ago, started that experiment and did the viability assays, and we have other assays that we want to test as well, and it's just the beginning of something. I can tell you that what's the cool thing about Denmark and Copenhagen is that we're super collaborative. You can see that with Zainab. She's literally like 10 minutes from our office space, and that is something, I come from Germany. We're always a little bit more suspicious and guarded there, I think. So for me, that's a new thing there, and I think that's the cool thing, that everyone is joining forces, and this is also what I'm looking here for, and I think that together people can really move a lot. So this is, I think, our approach as well for Neurophase Therapeutics. Yeah.
Great note to end on, Kim. Thank you very much. That was well said.